Hello and welcome back to my channel The Wise Pathogen. Today's topic is pure culture methods in microbiology. In today's session we will explore one of the most crucial techniques in microbiology that laid the foundation for the study of microorganisms, that is the isolation and cultivation of pure cultures. In this video we will cover the following points: pure cultures, their importance, history behind their development, key contributions from famous scientists like Joseph Lister and Robert Koch. Let us start by looking at how microorganisms actually exist in the natural world. In their native habitats, whether it's soil, water, plants, or even inside living organisms, microbes are rarely found in isolation. Instead, they live in dense, diverse communities composed of multiple species. These microbial communities are highly interactive, with organisms competing for nutrients, cooperating in metabolic processes, or coexisting in balanced ecosystems. For example, let us consider the human gut microbiome. It contains hundreds of different microbial species that interact with each other and with human cells. They help digest food, produce vitamins, and regulate the immune system. However, if we want to study just one specific bacterial species from the gut, these interactions pose a major challenge. Imagine trying to observe a single fish in a coral reef teeming with marine life. It is not at all easy. The same challenge applies to microbiologists who need to study individual microbial species. This complexity is exactly why microbiologists had to devise methods to isolate microorganisms from mixed populations that allows them to grow and study a single species in a controlled environment. To tackle this complexity, scientists developed a crucial concept that would change microbiology forever. That is pure culture. A pure culture refers to a laboratory culture that contains only one type of microorganism, no contamination, no competitors. Every cell in a pure culture originates from a single parent cell and is genetically identical to it. This level of control is essential for studying the physiology, behavior, and biochemical characteristics of microorganisms without interference from others. In clinical microbiology, for instance, when a patient presents with symptoms of infection, doctors need to determine which microorganism is responsible. This can only be done by isolating the pathogen in pure culture. Once isolated, scientists can study its properties such as antibiotic resistance or virulence more accurately. Pure culture also plays a critical role in industrial applications. The production of insulin, vaccines, and fermented foods like cheese or yogurt all rely on cultivating specific strains of microorganisms in isolation. For example, E. coli is used in biotechnology to produce human insulin, while yeast is central to brewing and baking. But why are pure cultures so vital? What makes them the cornerstone of microbiological science? Let us explore their true significance. The ability to work with pure cultures is foundational to modern microbiology. By studying one microorganism at a time, scientists can identify how it grows, how it interacts with drugs, and how it causes diseases. Let's take antibiotic resistance for example. To understand how a bacterial strain resists a certain drug, researchers must first isolate it in pure culture and expose it to a different antibiotic under controlled conditions. Pure cultures are also key in environmental and ecological microbiology. They also allow researchers to study how certain bacteria interact with their environment or other organisms. For example, rhizobium species form nodules on the roots of legume plants and fix atmospheric nitrogen. Hence, understanding this process requires isolating rhizobium in pure form. In research, pure culture provides a consistent, controlled environment. This allows scientists to trace metabolic pathways, observe genetic behavior, and experiment with gene editing, something that would be impossible in a mixed microbial population. However, getting to this point was not easy. Early microbiologists faced significant hurdles when trying to isolate pure cultures. Let us rewind to a time before these techniques were developed. Before the late 19th century, isolating a single species of microorganism was extremely difficult. Any contaminant, airborne or from equipment, could ruin the experiment. Furthermore, scientists did not fully understand the importance of sterilization. Without sterile instruments or clean techniques, contamination was nearly inevitable. Microbial colonies would grow as indistinct masses in liquid media, preventing researchers from isolating and identifying specific species. The lack of a solid medium also hindered progress. Without a surface for colonies to grow separately, microorganisms could blend together, masking their individual traits. That all began to change in 1878 when a British surgeon named Joseph Lister introduced a groundbreaking technique that changed the future of microbiology. Joseph Lister, already known for pioneering antiseptic surgery, also made major contributions to microbiology. In 1878, he developed the method of serial dilution, a technique that allowed scientists to isolate individual bacterial cells. In this method, a sample containing a mixture of bacteria is diluted step by step in sterile solutions until it is so diluted that only a few or even single bacterial cell remains in a given volume. This diluted sample is then introduced into sterile liquid media such as milk or broth. The lone bacterial cells grows and multiplies, forming a pure culture. One of Lister's earliest successes was isolating Bacterium lactis, a bacterium involved in milk fermentation. His work demonstrated that it was indeed possible to cultivate bacteria in isolation, an achievement that would inspire generations of microbiologists. Robert Koch revolutionized bacterial culturing that laid the groundwork for modern techniques. At first, Koch attempted to culture bacteria on the surface of boiled potatoes. This was one of the earliest solid surfaces used in microbiology. However, the results were inconsistent. Some bacteria wouldn't grow well on the potato, while others grew up unpredictably. To observe bacterial cells more clearly, Koch also began smearing bacterial samples onto glass slides and using dyes to stain them. This technique, now known as simple staining, helped improve visibility under the microscope. It was a great tool for visualization but not for culturing or growing bacteria in isolated form. Realizing he needed a more controlled environment to grow bacteria, Koch began working on liquid culture media. He used meat extracts and protein digests, materials that closely mimicked the nutrient composition of body fluids. This supported better bacterial growth, but liquid media presented a new problem, that is, bacteria would grow all throughout the broth, making it nearly impossible to separate or study individual species. To overcome this, Koch experimented by adding gelatin to the broth to solidify, creating a semi-solid medium. Koch then mixed bacterial samples into liquefied gelatin, poured them into shallow dishes and allowed them to cool and solidify. As the medium solidified, something remarkable happened. Individual bacteria stayed where they landed and began to multiply in place. The result: tiny visible colonies appeared, each one derived from a single bacterial cell. For the first time, microbiologists could observe and distinguish individual bacterial colonies growing on a solid surface. This was a monumental leap forward. For the first time, bacteria could be separated and studied in isolation. Each visible colony on the gelatin plate contained millions of genetically identical bacterial cells. This allowed Koch to isolate pure cultures of specific bacteria. He could then transfer these colonies into new media and grow them separately. This ability to isolate and propagate pure bacterial species opened the door to understand how specific microbes cause specific disease. It advanced microbiology from a largely observational science to an experimental one. The development of a liquefiable solid culture medium, in this case gelatin, was a revolutionary step in microbiology. It enabled precise identification, study, and replication of bacterial species, forming the methodological foundation we still use today. This discovery made it possible to diagnose diseases, understand microbial behavior, and even develop antimicrobial treatments. Gelatin was the first major solidified agent used in microbiology. When mixed with nutrient broth and cooled, gelatin formed a gel-like surface that could support microbial growth. This allowed researchers to observe individual colonies, each originating from a single cell. However, gelatin had serious flaws. Many bacteria could digest gelatin, breaking it down into liquid, which led to the loss of the solid surface. Gelatin also melted at around 28 degrees Celsius, a problem since many human pathogens grow best at 37 degrees Celsius. Despite its limitations, gelatin was a step forward, linked at the potential of solid media, but a better alternative was needed. And that better alternative came from an unexpected source, a kitchen recipe, and the ingenuity of a woman, Fanny Hesse. Fanny Hesse, the wife of microbiologist Walter Hesse, suggested using agar, a jelly-like substance derived from seaweed, as a solidifying agent. She had used it in the kitchen to preserve jams, and it did not melt easily or get digested by microbes. Agar offered two key advantages over gelatin. First, most bacteria cannot break it down, so it stays intact. Second, it remains solid at much higher temperatures, up to 100 degrees Celsius, making it ideal for culturing heat-loving microorganisms. Once introduced to microbiology by Hesse and adopted by Robert Koch, agar revolutionized microbial culturing. It became the standard medium for streak plating, colony isolation, and countless other techniques that are still in use today. Speaking of Robert Koch, his laboratory played a critical role in formalizing these methods and introducing even more tools that are still foundational today. Robert Koch, often regarded as the father of bacteriology, built upon Lister's and Hesse's work to create a complete toolkit for isolating and studying pure cultures. His lab developed nutrient broth and nutrient agar media that supported consistent bacterial growth. Most notably, Koch invented the Petri dish, a shallow container designed to hold solid media. This allowed for the growth and observation of bacterial colonies on a flat surface, with each colony arising from a single cell. Using these tools, Koch and his team were able to isolate and identify the organisms responsible for numerous diseases. Their work laid the groundwork for Koch's postulates, a series of criteria used to establish a causal relationship between a microorganism and a disease. One of Koch's most iconic achievements using these tools was the discovery of the bacterium responsible for tuberculosis. 1882, Koch made a groundbreaking announcement. He had discovered the bacterium that causes tuberculosis, a disease that claimed millions of lives. Using his newly refined pure culture techniques, Koch isolated Mycobacterium tuberculosis from patient samples. He then demonstrated through experiments and observations that this specific microorganism was responsible for the disease. This was a defining moment in microbiology. It showed that diseases could be traced back to specific pathogens. Koch's discovery also validated his postulates and set the standard for identifying the microbial causes of infectious diseases. While Koch's work focused on bacteria, a new frontier was beginning to emerge, one that involved agents even smaller and more elusive than bacteria, that is, viruses. In 1884, Charles Chamberlain, working with Louis Pasteur, developed a porcelain filter capable of removing bacteria from solutions. This filter would later prove instrumental in the discovery of viruses. Dmitry Ivanovsky and Martinus Behring used Chamberlain's filter to study tobacco mosaic disease in plants. They discovered that even after filtering out all bacteria, the plant sap remained infectious. This suggested the presence of a new type of infectious agent, much smaller than bacteria and not visible under the microscopes of the time. Behring called it a contagium vivum fluidum, a contagious living fluid. This marked the beginning of virology and opened a new world of research. From bacterial colonies to invisible viruses, the journey of microbiology has been built on the foundation of isolating microorganisms. Let us wrap up with a summary of what we have learned. As we reach the end of this journey through microbial history, one thing becomes clear. The development of pure cultures was a turning point in science, arguably one of the most important advances in biology and medicine. From Lister's serial dilution to Koch's Petri dish and the revolutionary use of Agar, each milestone helped scientists even more and navigated them closely into the microbial world. These innovations allowed us to identify pathogens, understand diseases, and develop life-saving treatments. Pure cultures gave us the power to isolate and study individual microbes, an ability that led to the discovery of antibiotics, vaccines, and even genetic engineering. The same techniques developed in the 19th century are still used in laboratories around the world today, now enhanced by molecular tools like PCR, genomic sequence, and CRISPR. Even in the era of metagenomics and microbiome research, where we study entire communities of microbes, pure culture method remains essential. Why? Because understanding how an individual microorganism behaves, how it responds to drugs, interacts with hosts, or survives in extreme environments require isolation and control. Whether we are battling new pathogens like COVID-19 or harnessing microbes for sustainable energy production, the foundational concept of the pure culture continues to drive progress. As so, through a single isolated cell, we gain insight into entire ecosystems. Pure culture techniques transformed microbiology from a chaotic into precise science, and their legacy continues to shape our world today.